Okay, guys. So today we're going to talk about Chapter 8, which is blood and blood splatter analysis. So part of crime scene investigation is to actually look for and locate any blood evidence, and then if there is any blood evidence that's found, to analyze that evidence. Now, blood stains can come in a variety of shapes, sizes, locations. There could be a lot, or it could be something that's very minute. Blood analysis is guided by the same laws of motion and forces that affect any liquid, meaning that there's a lot of physics that can be involved here. I don't want to dive too much into the actual physics behind this, but as you can imagine, the way that liquid sort of flows, blood will also flow. And that could also include splatters. And we'll look at some blood splatter patterns a little bit later in this presentation. So blood splatter patterns can determine the direction that blood is traveling. So bloodstains are something that are found at crime scenes. And as I mentioned a few minutes ago, they can vary in size and shape. A smaller droplet of blood can be more significant than a large pool of blood because that can actually tell a lot about what maybe happened versus just a pool that is just sitting there. Shape of the blood stain is affected by the force propelling the blood, meaning was there something that sort of an impact that led to that droplet or led to that splatter or led to even the pool. Blood analysis is guided by the same laws of motion that would affect any liquid. Uh, Using blood splatter pattern evidence, it is possible to demonstrate the direction the blood traveled, the relative force that was used, the angle of impact, and the area of origin, meaning where did this originate from? The size of the blood droplets is found in the blood splatter, provides evidence as to the type of weapon that may have been used to inflict the injury. So just before we kind of dive into actual blood analysis, let's just kind of look at some historical aspects here. This goes all the way back to 2500 BC, where Egyptians used bloodletting to cure disease. 500 BC, Greeks distinguished between arteries and veins. Arteries are going to be blood vessels that carry blood away from the heart. Veins will carry it back towards the heart. Then we kind of fast forward another 2,000 years, and this is where we get Carl Landsteiner, who discovered there are three primary blood types, A, B, and O. And these blood types are based off of the genetic factors that you inherit and that are expressed on your red blood cells. We have two primary antigens, A antigen and B antigen, and you may have one, the other, or both, or none. And depending on which antigens you are expressing on your red blood cells, that will determine your type of blood. This became important because we know that if an individual is given the wrong type of blood for transfusion, that can have very severe consequences. In 1971, so about 7 years later from the discovery of the blood types, forensic institutes began to train law enforcement on blood analysis and also collecting blood samples. This really came into light in 1995 with the O.J. Simpson trial. And then in 2008, followed by 2017, this is where we really started to develop guidelines for blood splatter analysis. So prior to DNA profiling, which is really started back in the late 90s, blood type analysis helped to identify an individual or link a person to a crime. The problem with just using blood type is that there are many people that have the same type of blood. Now, if you have some suspects and out of all those suspects, one of them matches a blood type that is significant, but it's not an absolute. Okay. Today, we do DNA profiling, which means we look at the DNA analysis. DNA is very unique to an individual. So if there is blood that is found in a crime scene, if they are able to isolate DNA from that blood, that is pretty much an absolute tie to an individual if it is a match. So today, DNA analysis has replaced blood typing for personal identifications. Now, before we start really diving into what is blood, what blood splatter analysis is and all of this, let's talk about what blood is. Blood is a tissue that is in your body. It is actually a liquid tissue, and it is a circulating connective tissue. And it consists of three main components. You have red blood cells, which are referred to as erythrocytes. White blood cells, there are many different types of these, but they're typically involved in immunity. And then platelets, these are cell fragments that are involved in blood clotting. Going back to the red blood cells, these red blood cells are what have the antigens present on their surfaces that will allow us to determine blood type. Now, during the maturation process of red blood cells, they do expel their nucleus. And that is important because if they expel their nucleus, they are also expelling their DNA. So red blood cells technically do not have any DNA in a mature red blood cell. So where do we get the DNA evidence from? It's going to be the white blood cells. The white blood cells are still whole cells in the sense that they still have a nucleus, so they will still have their chromosomes, they will still have their DNA. Okay, so that's where you're getting the DNA evidence from. White blood cell counts and immune response. The immune system identifies cells or molecules that are foreign. It recognizes viruses, bacteria, fungi, parasites, and proteins. And it sends white blood cells to the location to deal with any infections. We really don't focus too much on the white blood cells, except for the fact that this is where the DNA comes from. Blood typing. We have A and B antigens that are found on the surface of some red blood cells. If you are type O blood, you do not have either antigen. If you have the A antigen, you have type A blood. If you have the B antigen, you have type B blood. If you express both antigens, A and B, you're a type AB blood. And if you have no antigens, you're a type O blood. There's another factor that we actually look at, or another antigen, and that is what we call the Rh factor. And you either have it or you don't. If you have it, it will be designated by a positive sign next to your blood type. So like A positive. If you don't have it, it would be represented by a negative by the blood type. So for example, there, AB negative or O negative. These blood types are important because it really exhibits an antigen antibody response. Okay. What this means is that if you're given the wrong type of blood, let's say you're type A blood, but you're given type B blood, your body will react to that. And what will happen is it will cause clumping of the red blood cells, which is not good. And that can be fatal. So it is very important that, for transfusions, that blood typing is something that is accomplished. Now, looking at the probability in blood types, this is the frequency of the different alleles or variation in the genes that can exist. We can determine the probability that a particular blood type will appear within a particular population. Okay. And, in your textbook, it does go over the percentages of the U.S. population that have each of the four primary types of blood. Type O is 43% of the population. Type A is 42% of the population. Type B is 12% of the population. And type AB is 3% of the population. And again, these numbers are in your textbook there. So blood typing is not something that's technically used in forensics these days. It can be used as a quick test, this sort of narrow suspects. But for anything to sort of hold up in a trial or court as evidence, you really need the DNA evidence, which would definitively link a suspect to a crime scene. So characteristics of blood evidence. We do have forces affecting blood splatter. This could be cohesion, adhesion, and arterial spurt or gush. So if we look at the directionality of blood, you can have what's called a passive drop. This would be like if you're walking and bleeding and it's just dropping down from your body. The blood velocity and splatter size with this, you would have fine miss spatter pattern. You could have medium velocity size blood splatter or low velocity size blood spatter. We also do have very common blood stain patterns. This would be falling, projected, or smeared blood. Investigators can look at these blood stain patterns and interpret and even at times reconstruct what actually happens. There is something called the area of convergence. This is the estimation of the approximate location. Straight lines to the center of a long axis. If you draw the lines opposite of the direction, draw a small circle around where all the lines intersect, this would be your area of convergence. So if you kind of look at these droplets here, you can see that all of these lines kind of intersect right about here, okay? And this would be your area of convergence. The angle of impact calculations. Acute angle is formed when blood strikes a surface. With this, looking at the angle of impact, there are some calculations to sort of figure out with this. When you look at the angle of impact, you want to measure the width and the length of each droplet of the blood in millimeters. We use the metric system in science, so that's why we're using millimeters. Do not include any extensions of the blood droplet, such as a tail or a spine that might kind of spatter off of the droplet. The top and the bottom of the main droplet of the blood should be symmetrical. You want to divide the width by the length. This will give you a decimal value, meaning that this value will be less than 1. And then what you want to do is take the arc sign or inverse sign of the ratio width divided by length to determine the angle of impact. Once you have that angle of impact, what that can do is it is the acute angle that's formed when blood strikes a surface. When blood strikes a surface from a different angles, the blood stains vary in shape from round to progressively more elliptical. If blood strikes a surface at a 90-degree angle of impact, meaning that blood fell straight down, with only gravity and air resistance affecting the blood, then the shape of the droplet is round, having the same width and length. As the angle of impact decreases from 90 degrees to 10 degrees, the blood stains shapes become less round, and ultimately they'll become more elliptical in shape. When crime scene investigators process a crime scene, they can calculate the angle of impact based on the shape and length and width of the blood droplets. And this kind of gives them, once they have that angle of impact, it'll give them sort of information on where the blood originated from, meaning, was somebody hit in the head and this is a splatter that sort of came off of that. This could actually allow them to figure out where that impact with the head occurred. Now, the area of origin, um, this is based on the area of convergence and impact angles. Once the area of convergence and impact angles are determined, it is possible to estimate the area of origin, meaning, for example, if somebody was hit in the head with a hammer, where did that actually occur? Recall that the area of convergence indicates the source of the blood, but only in 2 dimensions. The area of origin goes one step further and provides a source of the blood in 3 dimensions. This can be calculated using a protractor and a string or laser pointer method, or you can use basic trigonometry to estimate the area of the origin. There are several steps that are outlined that will provide an estimate to the area of origin using the string method. And this is what a lot of crime scene investigators will do is they will use strings or lasers today to sort of determine the area of origin. And this really helps them to recreate what happened in a 3 dimensional aspect. Now, when at a crime scene and you do a walkthrough and information is gathered, you must take care not to disrupt any blood evidence. Blood evidence may not be as obvious as a pool of blood laying on the floor. Blood evidence might be a tiny droplet underneath the underside of, for example, in this picture, maybe the stove. Crime scene investigators need to gather information prior to collecting the evidence. Once the evidence is collected, you need to have proper documentation, proper methods of collecting, and there are reagents that can be used that can help to illuminate any blood that might not be visible with the eye. For example, luminol is something that you can use where if you are kind of looking in a room and you really don't see any blood per se, doesn't mean it's not there. It may have even been cleaned up, but there's still going to be traces. And what happens is if you use something like luminol, you can use a UV light and it will sort of light up areas where blood may have been. Once you do find a blood sample, the one thing you want to make sure is that it is that of human blood. And there are quick assays to determine that. Collecting blood evidence at a crime scene, you want to package it separately, make sure it's properly labeled. It's collected in a clean, unused, plastic sealed containers. Dry blood is packaged into paper bags, and wet blood stains are appropriately packaged as well. Especially if it's still a wet blood stain, you do not want to make sure, you want to make sure that you're using the appropriate packaging for that, as to make sure that A, it doesn't get contaminated, but B, that you don't end up with things like mold or other issues that could result. So a bloodstain analyst, this could be somebody in law enforcement, or it might be somebody on the lab side of the house. Formal training in blood pattern analysis is required. There's competency training, competency testing, as well as continuing education. There's always new information that's coming out. There's always new studies that are coming out. There's always new techniques that are being tested. So you want to make sure that if you are a person in this area, that you actually have uh, up-to-date information on what you're accomplishing there. Okay. So blood pattern analysis can be broken down into three primary stages here: pattern analysis, crime scene reconstruction, and consistency of scientific evidence with accounts of the crime. So when we look at pattern of analysis, this is the examination of the physical characteristics such as size, shape, distribution, and overall appearances. The analyst will interpret what patterns are present and what actions could have resulted in the blood patterns present. A specialist trained in blood pattern analysis may do the same, may do this at the crime scene. If a trained specialist is not at the crime scene, then the analyst could complete this in a forensics lab using images and evidence collected at the crime scene. And what they will try to do at that point is try to reconstruct what actually happened. So if we actually look at now crime scene reconstruction, this is based on the physical evidence found at the crime scene, such as the angles of impact, the areas of convergence or area of convergence, the areas of origin, transfer patterns, swipes, wipes. Wipes would be smear blood patterns created by an object moving through partially dry blood. Cast off patterns. For example, if a hammer was used and then that hammer was swung back, that could actually create what we call a cast off pattern. Um, and the questions that they want to ask with a crime scene reconstruction is what type of crime occurred, what type of weapon or weapons may have been used, how many instances of bleeding occurred, was this one, was this multiple times, is the blood human? That is something you want to check right off the bat. How many people were bleeding? Who was bleeding? Was it the victim? Was it the perpetrator? Was it both? Where on the victim or perpetrator's body was the source of the bleeding? How long has the body been there? Or if there's no body, how long has the blood been there? Is there evidence of movement of the victim or the suspect by the suspect? And then are there any transfer patterns found in the blood that could be used to identify the person that may have been at the crime scene? So once you sort of reconstruct the crime scene, then you want to make sure that you have consistency of the scientific evidence with the accounts of the crime. This will determine if there were any eyewitnesses that may have been present that could add some consistency to this. Determine if the injuries occurred are consistent with the weapons, tools that were used or suspected to be used. And can a suspect be excluded based on blood evidence found at the crime scene, meaning wrong blood type, um, or not human blood, or uh, ultimately DNA evidence would be your go-to there. The activities at the end of the um, investigation hopefully will answer all of these questions. Once in the lab, they can do various types of chemical testing. One, to confirm that it's human blood. There are some quick assays that can determine that right off the bat, even at the crime scene. These are typically what we call enzyme-linked immunoabsorbent assays, or ELISAs. An example of ELISA would be, for example, like the COVID test, where you kind of take a sample, mix it with a few things, run it on some type of diagnostic tool, and then you can end up with a result. Other testing can be done, such as toxicology. Is there anything in the blood? Alcohol, drugs, poisons, and then pathology is something that can also be looked at. Are there any abnormalities of the blood? Okay. That can also be inferred. One of the things that we do use in blood analysis is something called the Raman spectroscopy. This is a handheld device that's used at a crime scene. It determines if it's human blood. It estimates when the blood was deposited, meaning gives a timeline on how long the blood has been there. And most importantly, because it uses light, it does not destroy the sample. You could also use a 360-degree camera system. This will document the crime scene. It can be presented at trials. It calculates impact angles for you, and it helps to determine the area of origin. This is also, again, very importantly, documenting the crime scene, because at some point the crime scene will not exist. At some point the crime scene will be cleaned up, and the trial could be a year, 2 years, 3 years down the road. So you want to make sure you have adequate documentation on everything. You can also use unmanned drones to document, photograph, and video crime scenes. They can produce high resolution images, and this is used with additional software to make 3D images or to even reconstruct 3D versions of the crime scene. So, just sort of in summary, blood consists of cellular components and plasma. Blood provides both class and individual evidence. And forensics blood analysis was advanced through the work of several people here. It is something that is constantly evolving. Technologies are constantly evolving. And it's something that the analyst would need to keep up on. Blood types exist. The different types of blood types would be, again, A, B, O, or AB, and you can either be positive or negative for any of those four types. This results from the presence of antigens that are found on the surface of red blood cells. Blood stains can differ in size, shape, and patterns. Forces that can affect the falling of blood, and then blood splatter patterns or clusters of blood stains provide investigators with evidence. The angle of impact of a blood stain can be determined. The source of the blood can be determined using several well-defined drops of blood. And the area of origin can be estimated using string method, lasers, and basic trigonometry. So the basic trigonometry is just math to kind of figure out some of this stuff. Back in the day, they used to use strings. That's more of your low-tech version. Today, we have lasers that can sort of also do this. And we have cameras that can actually sort of, using the lasers, really pick up a lot of this and reconstruct maybe what happened. Care must be taken at a crime scene to avoid contamination. You want to make sure that you're not contaminating the blood source or even the DNA that's present, because that could actually cause issues longer down the road. Investigators will use chemicals such as luminol to locate blood. It's important that when you use chemicals, that you use chemicals that are not going to damage the samples. And new advancements in technology have improved blood evidence collection and analysis, and they are forever changing. Okay, that is it. For this chapter, there might be some calculations that you might need to do. Those formulas are in the text. The primary one is the one that we talked about earlier today in this lecture. Have a good one.